start our discussions on one of the major control systems of the body, the one probably we're most familiar with, the nervous system. We're going to start with, it's a very complex system, so we're going to start first with just nervous tissue itself. And then we'll spend the next few weeks going over different parts of the nervous system. So both the endocrine system and the nervous system are the systems that control our internal conditions. The endocrine system communicates by means of chemical messengers that we call hormones. Those messengers are secreted into the blood, and they will have an action on cells that have receptors for them. The nervous system uses cells called neurons to send messages from one to another, and they use an electrical and chemical system. They get a stimuli from an external environment or from another nerve. It makes its way to the central nervous system. The central nervous system processes the information and determines a response. And then the central nervous system can issue a command to muscles and glands to carry out that response. There are two major anatomical subdivisions of the nervous system. The central nervous system is the brain and spinal cord, while the peripheral nervous system are what we call the nerves and the ganglia of those nerves. A nerve is a bundle of nerve fiber axons wrapped in a connective tissue layer, and a ganglion is more of a swelling within a nerve where there are several nerve bodies held together, found in the peripheral nervous system. They kind of look like a knot. Functionally, there are also divisions of the peripheral nervous system. It's functionally divided into a sensory and motor division, and each of those has a somatic and visceral subdivision. So the sensory division is going to carry signals to the central nervous system from the sense organs. This is the afferent division. And if those signals come from receptors in the skin, muscle, bones, and joints, they are giving us somatic sensory information. So that's the somatic sensory division. Whereas if they're carrying signals from the heart, lungs, stomach, urinary bladder, other parts of the viscera, they are our visceral sensory division. The motor division is the efferent division. It carries the signals from the central nervous system to the effectors, which are many times muscles, which is where we probably got the motor from, but they also include glands, and they are divided into a somatic and visceral division. The somatic motor division carries the signals to skeletal muscles, causes voluntary muscles to contract, and it's responsible for our automatic reflexes. The visceral motor division is the autonomic nervous system, the ANS. It carries signals to glands, cardiac, smooth muscle, no voluntary control, and its responses are called visceral reflexes. There are sympathetic division and a parasympathetic division to the autonomic nervous system. Sympathetic division stimulates and prepares the body for action, while the parasympathetic division tends to have a calming effect on the body. We also have a group of nerves within the digestive tract that coordinate communications within the digestive tract called the enteric plexus. So we have the central nervous system and the peripheral nervous system. Central nervous system divided into the brain and spinal cord. The peripheral nervous system is divided into two divisions, the sensory division and the motor division. Those are both divided into a visceral and somatic division, and the motor division of the peripheral nervous system is further divided into the sympathetic division and the parasympathetic division primarily. We will also see the enteric plexus later on when we talk about the digestive system. Neurons have the ability to communicate with other cells because they are excitable. Again, that means they can respond to stimuli. They are conductive. They produce electrical signals that are conducted down their length and cause things to happen in other cells. And they are secretory. They can, when that signal reaches the end of an axon, the neuron will release its communicating molecule, the neurotransmitter, to stimulate the next cell. There are three functional classes of neurons. They can be sensory neurons, part of the afferent division, bringing things to the central nervous system. They begin in every organ of the body and terminate in the central nervous system. Vast majority of neurons are interneurons. They receive signals from neurons, process the information, and make decisions about whether to stimulate the next neuron in line or not. These interneurons lie entirely within the central nervous system. They're connecting the motor and sensory pathways. Again, this is the most functional, most common functional type, and about 90% of all neurons are interneurons. And motor neurons, or efferent neurons, send signals out to the muscles and glands, the effectors. The principal components of a neuron include the cell body. It's surrounded by it's also called the soma, the neurosoma on occasion, the perikaryon, which means around the nucleus. The cell body is where you find the nucleus and many organelles. There will be mitochondria here, lysosomes, Golgi complex, inclusions if they're there, rough endoplasmic reticulum. Its cytoskeleton has the microtubules and the neurofibrils, and it's used to compartmentalize the rough ER into dark staining chromatophilic substance. There are no centrioles. Mature neurons cannot undergo mitosis after adolescence. Those inclusions may be glycogen granules, lipid droplets, melanin, lipofuscin, which is a pigment resulting from the degradation of organelles by lysosomes. They have neurites, or extensions, that reach to other cells. Dendrites are the most numerous of the neurites. They resemble branching trees. They're primarily the sites for receiving signals from other neurons. Neurons can have one dendrite or thousands of dendrites. The axon, or the nerve fiber, is a long cylindrical extension, relatively unbranched, but gives off many axon collaterals that are specialized for rapidly conducting nerve signals. They originate at the cell body location known as the axon hillock. They contain axoplasm. It's cytoplasm, but it's found in the axon, and we call the membrane around it an axolemma. At its end, axon does branch profusely, so even though they only have one axon, that one axon makes contact with potentially many target cells. Each branch of arborization ends in a bulbous axon terminal, which forms a synapse with the next cell that we'll get into. A neuron never has more than one axon, but some neurons actually do not have any axons. So generally speaking, we have the cell body here, dendrites bringing information into the cell body, and an axon, which carries information away from the cell body. And here is one stained microscopically. There are 4 structural classes of neurons that are determined by the number of cell processes extending from the cell body. We can have a multipolar neuron with one axon and multiple dendrites. This is probably the most common type in the body. Most of the neurons in the central nervous system are multipolar neurons. We could have a bipolar neuron, which has one axon and one dendrite. We find these in olfactory cells in our nose, some neurons in the retina, sensory neurons in the ear. These are going to be found mostly in our special senses that we will get to later on in the semester. We can have a unipolar neuron, which is a single process leading away from the cell, but it quickly splits into peripheral process and a central process. Both processes comprise the axon. There's only a short receptive ending on the peripheral processes that are considered dendrites. An axonic neuron has many dendrites but no axon. We find these in the brain, the retina, and the adrenal gland. Axonal transport is the two-way passage of materials along an axon. Some materials made in the cell body must be transported to the axon terminal, things like enzymes and ion channel proteins. We can get that would be known as anterograde transport, which would be movement away from the cell body down the axon, driven by a motor protein called kinesin. There are other materials needed to be transported from axon terminals back to the cell body, and this would be retrograde transport. Axon towards the cell body, driven by the motor protein dynein. The rate of axon transport can be fast or slow, with fast axonal transport, which would be 200 to 400 millimeters per day. Think about that. That can be anterograde or retrograde. Slow axonal transport is 0.2 to 0.5 millimeters per day. That's a stop and go movement. It results in the slower speed. That is always anterograde. This is how we transport enzymes, cytoskeletal components, new axoplasma. Damaged nerves regenerate at a speed governed by this slow axonal transport, 0.2 to 0.5 millimeters a day. Approximately 86 billion neurons in the adult brain, and a roughly equal number of neuroglia, or glial cells, that are non-neuronal supportive cells. These cells have the functions of binding neurons together, forming a supportive tissue framework. In a fetus, they guide migrating neurons to their destinations. They also cover mature neurons, except at the synapses, we'll get into a little later. When that covering prevents neurons from touching each other directly, gives precision to conductive pathways. The four types of glia that occur in the central nervous system include the oligodendrocytes, which form the myelin sheaths in the central nervous system, the epidermal cells, which line the internal cavities of the brain and secrete circulating cerebral spinal fluid, the microglia, which are a type of macrophage that engulf debris and provide defense against pathogens, and the astrocytes, the most abundant type, with the widest variety of functions. They may provide a framework for nervous tissue. They have extensions that contact the capillaries and stimulate formation of the blood-brain barrier, help to adjust blood flow, supply neurons with lactate for energy, secrete nerve growth factors, influence synaptic signaling, regulate the composition of tissue fluid, and form scar tissue when the neurons are damaged, a process called astrocytosis or sclerosis. There are two other types that are found in the peripheral nervous system, the Schwann cells, or more correctly, the neurolemnocytes. Schwann was the anatomist who first described them. They envelop the axons of the peripheral nervous system, forming a myelin sheath and assisting in the regeneration of damaged nerve fibers. There are also satellite cells surround the nerve cell bodies in the ganglia of the peripheral nervous system, and they do work similar to the astrocytes. They provide insulation around the cell body and regulate the chemical environment that those cells are in. Most brain tumors are actually glial cell tumors. Mature neurons really can't divide, so they seldom develop into tumors. But brain tumors can arise from the meninges, the protective membranes of the central nervous system. They could also metastasize from non-neuronal tumors in other organs, or they may be glial cells. Those glial cells are mitotically active throughout life. So a glioma is a tumor of a glial cell. They tend to grow rapidly, and they tend to be highly malignant. The blood-brain barrier decreases their susceptibleness to chemotherapy, and treatment generally consists of radiation or surgery. The myelin sheath is a spiral layer of insulation around an axon. In the peripheral nervous system, the myelin sheath is produced by Schwann cells, while in the central nervous system, oligodendrocytes make the myelin sheath. The sheath consists of the plasma membrane of these cells with 20% protein, 80% lipid. Production of the sheath is called myelination, begins during fetal development, proceeds rapidly during infancy, and then it is completed by late adolescence, early adulthood. A Schwann cell will spiral around repeatedly around a small section of a single axon, lays down as many as 100 layers of its membrane with no cytoplasm between the layers. These layers make up the myelin sheath. The outermost neurolemma is going to contain Schwann cell nucleus and most of its cytoplasm. It's going to be external to the neurolemma. It will be just under the basal lamina and the endoneurium, thin layer of connective tissue, as shown here. In the central nervous system, oligodendrocytes are going to provide the myelination, and they will provide several processes that will wrap around small portions of many axons in its immediate vicinity. So during myelination, the nucleus cannot migrate around the axon like the Schwann cell does. They must push newer layers of myelin under the older ones, so myelination will spiral inward toward the axon, and in this case, we do not have a neurolemma, as shown here. In both the peripheral and the central nervous system, the myelin sheath is segmented. There are many Schwann cells needed to myelinate a single axon, and in the central nervous system, although one oligodendrocyte is providing some of the sheath, it is many oligodendrocytes that are wrapping an individual axon. The gap between the myelin sheaths, the segments, is called the myelin sheath gap, traditionally known as the node of Ranvier. There are internodal segments, which are myelin-covered segments. Their initial segment is a bare section of axon between the axon hillock and the first glial cell. A trigger zone includes the axon hillock and the initial segment that will play an important role in initiating a nerve signal. Multiple sclerosis is a disease in which the oligodendrocytes and the myelin sheaths in the central nervous system deteriorate, and that myelin is replaced by scar tissue. That causes nerve conduction to be disrupted, leading to things such as double vision, tremors, numbness, speech defects. Typically, the onset is between 20 and 40, and it's generally fatal from 25 to 30 years after diagnosis. We are sure it is an autoimmune-based disease, and it's possible that autoimmunity is triggered by a virus. There is another disease of myelin sheaths known as Tay-Sachs disease. It's a hereditary disorder seen mainly in infants of Eastern European Jewish ancestry, although it is not uncommon in French Acadian ancestry. It is usually fatal before the age of 4, and it results from a genetic mutation that leads to the abnormal accumulation of a glycolipid called GM2 in the myelin sheath. There are also unmyelinated axons in the central nervous system and the peripheral nervous system. In the peripheral nervous system, Schwann cells hold small unmyelinated axons in surface grooves. The membrane folds once around each axon but doesn't spiral repeatedly, wrapping the nerve, serving as a neurolemma, and the basal lamina surrounds the Schwann cell along with its axons. And whether they have myelin around them or not is one of the things that will determine how quickly the speed of a nerve signal is, the conduction speed of the axons. Other things that will affect that are the diameter. Large axons have more surface area and conduct signals more rapidly. Myelin actually speeds up signal conduction. So we could have small unmyelinated fibers that would transmit their signal between 0.5 and 2 meters per second. Small, lightly myelinated fibers between 3 and 15 meters per second, and large myelinated fibers can fire at a rate of up to 120 meters per second. Regeneration of peripheral nerve fibers is possible if the nerve cell bodies are intact and some of the neurolemma remains. The steps for that regeneration include the axonal distal to the injury degenerating. The macrophages clean up the debris. The cell body will then swell. The endoplasmic reticulum will break off, and the nucleus will move off center. Those changes are due to the loss of nerve growth factors from the target cell. The axon stump will sprout with multiple growth processes. Then Schwann cells, neurolemma, endoneurium, and basal lamina will form a regeneration tube, and that tube will guide the regrowth of the axon to its original destination. Once contact is reestablished with the original target, the neurosoma cell body will shrink and return to its original appearance. The nucleus returns to its normal shape. The atrophied muscle fibers begin to regrow. Regeneration is not fast, it's not perfect, and it doesn't always work. It's a very slow process which can take up to 2 years. Some axons connect with the wrong muscle fibers, some don't connect with anything and die. Central nervous system axons generally do not regenerate. Neural communication is based on electrical potentials and currents. So back to physics class for a minute. We have electrical potential, which is a difference in concentration of charged particles between one point and another. That is a type of potential energy. The measurement unit for that is volts, and under some conditions that voltage can produce a current. Current is a flow of those charged particles from one point to another. So a flashlight battery has a potential of about 1.5 volts. It can produce a current of electrons that will light a bulb. With a potential voltage, the battery is polarized. Living cells are also polarized. The resting membrane potential is the charge difference across the plasma membrane. It's typically around minus 70 millivolts in an unstimulated or resting neuron. Again, that's negatively charged particles on the inside of the membrane, many of which are proteins compared to the outside. And then electrical currents in the body are created by the flow of ions, sodium ions, potassium ions, through gated channels in the membrane. The ionic basis of the resting membrane potential is that the ions are unequally distributed between extracellular and intracellular fluid. So the resting membrane potential results from the diffusion of ions down their concentration gradients through the membrane, the selective permeability of the membrane, meaning it allows some ions to pass more easily than others, and the electrical attraction of the cations and the anions to each other. Potassium has the greatest influence on the resting membrane potential. Potassium is more concentrated in the intracellular fluid compared to the extracellular fluid. The cell membranes are more permeable to potassium through the leak channels than other ions. Potassium leaks out inside of the membrane, becomes more negative, resulting in electrical attraction that will help bring potassium back in. You will reach equilibrium eventually. There will be no net movement of potassium when the tendency for potassium to exit equals the tendency for potassium to enter. It's exiting because of its concentration gradient. It's entering because of the negative charge, the electrical attraction. Sodium also has an influence on the resting membrane potential. Sodium, a positive ion, is more concentrated in the extracellular fluid compared to the intracellular fluid. But the membrane is much less permeable to sodium than it is to potassium. Sodium diffuses into the cell down its concentration gradient, and it is attracted electrically, but it's a relatively small amount. That cancels some of the negative charge, reducing the voltage across the membrane. There is a sodium-potassium pump that compensates for the continual leakage of sodium and potassium. It causes sodium to move out of the cell and brings potassium into the cell, maintaining their concentration gradients, about -3 millivolts of the resting membrane potential. And it works continuously, but it also requires ATP to function. That about 70% of the energy requirement of the nervous system is for this, sodium-potassium pump. Sensory neurons can be stimulated by different things, such as chemicals, light, heat, or mechanical forces. The stimulation will trigger a local temporary change in the membrane potential. So a chemical stimulant can bind to a receptor on a neuron that will open a sodium channel and allow sodium to come in. Entry of the positively charged sodium makes the membrane potential less negative. Polarity is reduced. Voltage is less negative. It's called depolarization. Depolarization spreads from the point of stimulation. This local spread is called the local potential. It's relatively short-ranged and temporary. We can get local potentials that are either graded, which means they vary in magnitude with stimulus strength. The stronger stimuli will open more sodium channels and they will stay open longer. It can be decremental and get weaker the further they spread from the point of stimulation. They can be reversible. If the stimulation ceases, the membrane voltage will quickly return to its normal resting potential. And so they can either be excitatory or inhibitory. Depolarization is excitatory, makes the neuron more likely to fire an action potential. The action potential is the signal. Hyperpolarization, making the membrane more negative, will be inhibitory. It makes the neuron less likely to produce an action potential. That action potential is the rapid up and down change in voltage produced by coordinating the opening and closing of voltage-gated ion channels. It will only occur where there is a high enough density of voltage-gated ion channels, the trigger zone of the axon, and if the excitatory local potential reaches the trigger zone and is still strong enough, it will open voltage-gated sodium channels and generate that action potential. So if we want that nerve to fire, the local potential has to trigger an action potential. The local potential spread to the axon hillock. The voltage at the axon hillock reaches a threshold. The minimum voltage to open voltage-gated channels is around -55 millivolts. Once that occurs, the voltage-gated sodium channels open quickly. Voltage-gated potassium channels open more slowly. Sodium enters and depolarizes the membrane, quickly going past 0 millivolts. Sodium channels become inactive and start to close. The voltage typically peaks around 35 millivolts by the time the inflow of sodium ceases. And that membrane polarity has reversed. Now it's more positive on the inside than the outside. The voltage-gated potassium channels will then be fully opened. Potassium will flow out of the cell and the membrane will become more negative again. It will repolarize. Potassium continues to exit and produces a negative overshoot. We get what we call hyperpolarization, more than 1 or 2 millivolts more negative than the original. And then the membrane voltage will return to the resting membrane potential as that sodium-potassium pump allows for leakage of the sodium and resets the process. Concentrations of sodium and potassium on either side don't significantly change during an action potential. The movement of only a few ions can have a large effect on membrane potential. Only about 1 in a million ions cross the membrane during an action potential. Only the thin layer of ions close to the membrane is affected. And even after thousands of action potentials, the cytosol still has a higher concentration of potassium and a lower concentration than sodium than the extracellular fluid. So some of the characteristics of the action potential is it is an all or nothing. All or nothing law states that if a threshold is reached, the neuron will fire to maximum voltage. If the threshold is not reached, it does not fire. It is non-decremental. You don't get weaker with distance. And it is irreversible. Once you start it, the action potential will travel all the way down the axon and it cannot be stopped. There is a refractory period, which is the period of resistance to stimulation. We can divide it into the absolute refractory period, where no stimulus of any strength will trigger another action potential. And this is caused by inactivation of the voltage-gated sodium channels. A relative refractory period is when, if you had an unusually strong stimulus, you might be able to trigger a new action potential. So during hyperpolarization, a larger depolarization is required to reach the threshold. And those are mapped out here. Unmyelinated axons and continuous conduction. So unmyelinated axons have a voltage-gated channel along their entire length. The action potential triggered at the trigger zone causes sodium to enter the axon and diffuse into adjacent regions. Depolarization will open voltage-gated channels. Opening of voltage-gated ion channels results in new action potentials, which then allows sodium to diffuse to excite the membrane immediately distal to that. That chain reaction will continue down the axon like a wave of falling dominoes, and this is called continuous conduction. You have to send the signal all the way down an unmyelinated axon. In a myelinated axon, we get what are known as saltatory conduction. The action potentials are generated at the nodes, where the voltage-gated ion channels are concentrated. The electrical signals spread passively between the nodes, but very quickly. The strength does decrease. When the signal reaches the next node, it's still strong enough to depolarize the membrane to the threshold. And so the sodium channels open and a new full-strength action potential occurs. So the action potential seems to jump from node to node, and therefore it moves faster down the axon. A synapse is a point where an axon terminal meets the next cell in the nervous system. It's a neuron, and the peripheral might be a gland or a muscle cell. So for neuron to neuron synapses, the action potential arrives at the end of a presynaptic neuron. Presynaptic neuron will release a neurotransmitter, and the postsynaptic neuron will respond to it. The synaptic cleft, or the gap between the neurons, was discovered by Kajal through histological observations. Louis, in 1921, demonstrated that neurons can communicate by releasing chemicals. He flooded the exposed hearts of 2 frogs with saline, stimulated the vagus nerve of the first frog, and its heart slowed, removed the saline from the first frog, and found it slowed the heart of the second frog. He named it the vagus substance. It was later renamed acetylcholine, and it was the first neurotransmitter to be discovered. Neurotransmitters are released at chemical synapses. There are also electrical synapses that occur between some neurons, neuroglia, cardiac, and single-unit smooth muscle cells. These are gap junctions that join the adjacent cells, and in this case, the electrical signal spreads directly from cell to cell. That's much faster. There's no delay for release, no wait for diffusion, no binding of neurotransmitter, but it makes integration or figuring out the information impossible. The synaptic cleft is about 20 microns wide. There are cell adhesion molecules reaching into the cleft. Those cell adhesion molecules link the two cells together. Axon terminals of presynaptic neurons contain synaptic vesicles that have the neurotransmitter in them. There are many vesicles docked on release sites on the plasma membrane, ready to be fired. A postsynaptic neuron contains higher postsynaptic density of neurotransmitter receptors and ion channels. These are ligand-gated ion channels that open when the neurotransmitter binds to them. Here is a micrograph. We see the presynaptic neuron connection and the postsynaptic neuron cell body. And if we looked at it from an artist's point of view, we see this. We have lots of vesicles ready to dump neurotransmitter and lots of receptors for those neurotransmitters ready to respond to that neurotransmitter. There are more than 100 neurotransmitters identified. Most of them fall into categories such as acetylcholine, which is formed from acetic acid and choline. Amino acids, they might be glycine, glutamate, aspartate, and GABA. Monoamines are synthesized from amino acids by removal of the carboxy group, but keep the amino group. These would include the catecholamines, epinephrine, norepinephrine, dopamine, and serotonin and histamine, or the purines, adenosine, and ATP. There are gases such as nitric oxide and carbon monoxide, synthesized as needed instead of being stored. Neuropeptides are 2 to 40 amino acids, CCK or cholecytokinin and endorphins are these type. They are stored in large secretory granules, and they also function as hormones or neuromodulators. So synapses are variable in their modes of actions. Some neurotransmitters are excitatory, others are inhibitory. Sometimes the transmitter effect differs depending on the receptor on the postsynaptic cell. Some receptors are ligand-gated channels, others act as second messenger systems. So 3 kinds of synapse we'll look at are the excitatory cholinergic synapse, the inhibitory GABA synapse, and the excitatory adrenergic synapse. So the cholinergic synapse is the excitatory. Action potential will depolarize the axon terminal, opening voltage-gated calcium channels. Calcium will enter, trigger the exocytosis of acetylcholine. That acetylcholine will diffuse across the cleft and bind to postsynaptic receptors. The acetylcholine receptors are ligated ion channels that open, allowing sodium and potassium to cross the membrane. Remember, it is the entrance of sodium that causes depolarization of the postsynaptic potential. If strong enough, the depolarization will spread to the trigger zone and raise the membrane potential to the threshold and trigger an action potential. In a GABA synapse, we have an action potential triggering the release of GABA into the synaptic cleft. The GABA receptors are chloride channels. Chlorine will enter, and that's got a negative charge, and it will hyperpolarize the postsynaptic membrane. And therefore, the postsynaptic neuron will be inhibited. It's less likely to fire because it's less likely to reach its action potential or its local potential. And the excitatory adrenergic synapse, we can use norepinephrine as an example. It's also called noradrenaline. The monoamines and neuropeptides bind to a G protein coupled receptor on the postsynaptic membrane. That activates a second messenger system, cyclic AMP, for example. This makes them a little slower to respond than cholinergic or GABA synapses, but it does have the advantage of amplification. A single molecule of norepinephrine produces vast numbers of second messengers within the system, and that's generally what I would like you to remember when you hear second messengers, amplifying the effect. It's important not only to stimulate the postsynaptic cell, but also to turn off the stimulus. The neurotransmitter stays bound to the receptor for about a millisecond. If the presynaptic cell continues to release the neurotransmitter, one molecule is quickly replaced by another, and the postsynaptic cell continues to stimulate. So to end the signal, first we need the presynaptic cell to stop releasing its neurotransmitter, and we need the neurotransmitter already in the synapse to be cleared. We can clear that using neurotransmitter degradation. An enzyme in the cleft can break down the neurotransmitter. For example, acetylcholinesterase breaks down acetylcholine into its choline and acetate. Reuptake, sometimes a neurotransmitter, brings that back into the cell, either the neurotransmitter itself or the breakdown products from the enzyme. Or simple diffusion, where the neurotransmitter breaks down, or its breakdown products simply diffuse away from the synapse into the nearby extracellular fluid. There are also neuromodulators. Neuromodulators are chemicals secreted by neurons that have long-term effects on groups of neurons. They may alter the rate of neurotransmitter synthesis, release, reuptake, or breakdown. They may adjust the sensitivity of the postsynaptic membrane. Nitric oxide is a simple neuromodulator. It's a gas that enters the postsynaptic cells and activates second messenger pathways to relax smooth muscle. Neuropeptides are chains of amino acids that can act as neuromodulators. Enkephalins and endorphins are neuropeptides that inhibit pain signals in the central nervous system. Integration is the ability to process, store, and recall information. It's used to make decisions. Chemical synapses allow for decision making. Brain cells are incredibly well connected, allowing for complex integration. There are pyramidal cells of the cerebral cortex that have about 40,000 contacts with other neurons. Trade-off for this is that the chemical transmission involves a synaptic delay, and that makes information travel slower than it would be if there was no synapse. There are two types of postsynaptic potentials produced by neurotransmitters: excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Different neurotransmitters cause different types of postsynaptic potentials. Glutamate and aspartate produce excitatory. Glycine and GABA produce inhibitory. A neurotransmitter might excite some cells and inhibit other cells, depending on the receptor in the postsynaptic membrane. Acetylcholine, norepinephrine both work in this way. Acetylcholine excites skeletal muscles, but it inhibits cardiac muscle, and that's due to the acetylcholine receptor on the different types of cells. Summation is the process of adding up all the postsynaptic potentials and responding to their net effect. This will occur in the trigger zone. So some incoming nerve fibers are excitatory while others are inhibitory, and whether or not a neuron fires will depend on whether its net input is more excitatory or inhibitory. And it's the balance between excitatory and inhibitory that allows information to be processed. Two ways excitatory postsynaptic potentials can be reached. We can get temporal summation or spatial summation. Neurons can work in groups to modify each other's actions. We can have a presynaptic facilitation when a presynaptic neuron enhances another one. Inhibition can occur when one presynaptic neuron suppresses another one. Neural coding is converting that stimulus information into a meaningful pattern of action potentials. We have a labeled line code, which is the mechanism for transmitting qualitative information, and quantitative information is encoded either by weak stimuli sensitive to low threshold or weak stimuli causing neurons to fire action potentials at a slower rate. Neural pools are functional groupings of neuron. Each pool consists of thousands of interneurons connected with a particular body function, controlling rhythm of breathing, moving limbs rhythmically when walking, for example. They will have discharge and facilitated zones. Information arrives in the neural pool through the input neurons. Within the input neuron's discharge zone, it can act alone to make postsynaptic cells fire. In a broader facilitated zone, the input neurons make fewer, less powerful synapses, so they can only stimulate targets with the assistance of other neurons. We can have diverging circuits that spread, or converging circuits that funnel. Reverberating circuits in which neurons stimulate each other in a linear sequence, but one or more in the later cells re-stimulates the first cell. We can also have parallel after discharge circuits in which the input neuron diverges to stimulate several chains of neurons. We can use serial processing in which neurons and neural pools relay information along a pathway in a relatively simple linear fashion. We can also have parallel processing in which the information is transmitted in diverging circuits through different pathways that act simultaneously or with a different purpose. A memory is a pathway of synapses through the brain. It's the physical basis of memory. Along the pathway, new synapses are created or existing synapses are modified to make transmission easier. So synaptic plasticity is the ability of synapses to change. Synaptic potential is the potentiation is the process of making the transmission easier. We have three basic kinds of memory: immediate memory, short-term memory, and long-term memory. They're correlated with different modes of synaptic potentials that last from a few seconds to a lifetime. Immediate memory is the ability to hold something in your thoughts for a few seconds. It's essential for reading, feel for the flow of events. Our memory of what just happened may depend on reverberating circuits. Short-term memory lasts from a few seconds to a few hours. We also include working memory in that. So calling a phone number you just looked up. Synaptic facilitation makes it easier to transmit signals across the synapse. It's important for memory formation. Long-term memory can last up to a lifetime and have more information than short-term memory. The long-term memory can either be explicit or declarative memory or implicit memory. Some long-term memory involves the formation or remodeling of synapses. We get new branching of axons or dendrites, and also some long-term memory involves molecular changes called long-term potentiation. We do forget, and long-term depression may account for some of that forgetfulness. Low-frequency stimulation results in lower levels of intracellular calcium. Alzheimer's disease is a neurodegenerative disorder. About 11% of the population over 65, 47% of the population by 85. You're probably familiar with many of the symptoms of Alzheimer's disease. It's currently only confirmed at autopsy, and we get atrophy of gyri in the cerebral cortex. There are new treatments coming out. This particular one, they describe, has been halted, but there are newer drugs, and the potential to treat Alzheimer is certainly improving. And you can see the brain on autopsy. Another is Parkinson's disease. This is a progressive loss of motor function due to the degeneration of dopamine-releasing neurons. This leads to involuntary muscle contractions, pill rolling motion, facial rigidity, slurred speech in a typical gait. There are drugs in physical therapy. Dopamine precursor, L-DOPA, can cross the brain barrier. It does have side effects. MAO inhibitors tend to slow neurodegeneration, and there are surgical techniques that can be used to relieve tumors. All right, I hope you enjoyed Veterans Day, and I will post something for the lab on Thursday.